Welcome to the Conversational Flow, a KCAS Bio podcast.
Hello, and welcome to the 25th episode of the Conversational Flow, an official podcast of KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. Unlike KCAS' other podcasts, the Weekly Bioanalysis, which features news and topics more generally associated with the world of bioanalysis, the Conversational Flow focuses more on the niche of flow cytometry and its role within the greater industry. My name is Adam, and I've been in the flow cytometry space for over 15 years. I'm here with my co-host, Brian.
Thanks, Adam. My name is Brian. I've been in the flow cytometry space for over 14 years.
We're joined by a special guest today. David, would you like to introduce yourself briefly?
Hi, my name is David Leclerc. I'm the director of the CAT facility at the University of Chicago. Thanks for having me.
Thank you, David. Looking forward to talking with you. Brian and I are members of the growing leadership and scientific advisory team at KCAS. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. While our KCAS headquarters are located in Kansas, with additional location in Europe, where Brian is currently, I myself are in Philadelphia. Jeremy, our producer, is in Missouri, and our special guest, David, is in Chicago. We're all truly all over the map. We're exhilarated to have you listening to our 25th episode of our podcast, which will be available virtually everywhere. Wherever you choose to find and play your podcast, you'll likely be able to find the Conversational Flow. We welcome all of you, whether you're joining us for the first time or if you're a regular listener to any of our other podcasts. Today's episode will be a review of the latest news and resources, and then a focus on a topic of our choosing before discussing any feedback we've received from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. So again, we're amped up to have you here and looking forward to a fun episode today. To kick us off for episode 25, Brian is going to go over news and resources before diving into today's topic. Brian?
Thanks, Adam. We've got a meaty agenda today.
Very good. I like the change there.
Right? And the reason I say that is, of course, because the first two pieces of medical research news actually both came with a paper, so a published academic journal article here. So the first one is around a new study that drew the link in between the protein quality control system and some of the checkpoint inhibitor failures that we see in cancer.
Yeah, and I'm totally going to divert just slightly, going to my background here. So I did my undergrad at Penn State. So seeing the lead of Ohio State, I think a couple of weeks ago, it probably would have been more bummed to see it. But the way the team Penn State has played lately, not too bummed. But anyways, fantastic article here that excited to talk about as far as stress responses linked to protein misfolding, right? And specifically here, we're talking about T cells, and they're going over this pathway, TEX-PSR. And so it's essentially a stress response pathway to protein misfolding. And instead of down-regulating protein synthesis, it actually accelerates it, which, what you're looking at here is T cell exhaustion. And why does that matter? When you have T cell exhaustion, it prevents the T cells, the immune system, from really combating cancer cells. And so, it's interesting because the protein misfolding and the issues that it caused had actually been controversial, it looks like, until about 2002. And since then, more and more has been understood about it. And we've even talked somewhat about protein folding, where it is as far as AI predictive models. And I believe that was a Nobel Prize, if I'm remembering correctly. Right, Brian?
It was. And, what's interesting about this is that I feel like with T cell exhaustion, it's always been sort of a known phenomenon where a lot of the immunotherapies that target cancers will seem to work great, definitely in vitro experiments, sometimes even in preclinical models. And then when it gets into humans, it's just not as effective as it was originally thought to be. in part because T cells, as they're combating in the tumor microenvironment, get exhausted more quickly than the modeling suggests they should. And this is now sort of giving a real mechanism behind why that may happen, which means if we know how it happens, then maybe we can block it from happening as well.
Right. And, you're talking about modeling. There's only so many things that you can take into account when you do modeling. And protein folding is dependent on the sequence, the amino acid composition, as well as environmental influences. And so when you're in a tumor microenvironment, that environment starts to become highly variable and so can really throw off some of those models. And so there, like you said, I think this whole pathway and understanding how it's impacting becomes very helpful in engaging those T cells, activating those T cells, and then generating therapeutics that take advantage of that.
Absolutely. Yeah, so excellent paper to start out with. And then the next paper I saw came out, and it came out from a number of the authors of the SoulCap initiative. And it also involved AI. I know that you also collaborated with a few of these authors on your recent paper, Adam. So that's what caught my eye about this one. So this is called AI in Flow Cytometry, Current Applications and Future Directions, published in Cytometry Part B.
Mm-hmm. It's funny when I saw the title, current applications, I'm thinking the future directions. Where wouldn't it be, right? I mean, right now we don't have AI running the samples for us, but there's so many different directions that you could apply AI and are specifically. One of the things that it brought to mind was at ICCS last year, 2024, so this was in Seattle, and went through a whole effort of using AI to go from building a validation to writing a plan to generating worksheets to writing a report, analyzing data, all of that in the span of 20 minutes. And so it's just an incredible demonstration of how AI can really pull things together much more quickly and enhance efficiency. And of course, the warnings all still apply, double checking, making sure that the output is appropriate. But here talking about, it's always applied now to analysis. That's one of the bigger things the industry is moving towards, automated gating and really embracing that, as well as, you can use it in panel design and study design, things of that nature. What are your thoughts there, Brian?
Yeah, and I think what is most exciting to me is that you're absolutely right that I'm so used to seeing, AI is the future. This is what will happen in Flow because of AI in just a few short years. And now this is talking about what is currently happening. And that's very exciting to me because it's actually translating into faster results, more robust checking of different aspects, more consistent checking of different aspects. And yeah, it's just being applied in a lot of different contexts. So kudos to the authors to bringing it together and summarizing it for the field, and really drawing attention to the fact that this is now considered not just a nice to have or a nice to think about in the future, but a must have for best practice.
Now, I got my finger on a question here just because of the conversations that you and I always have internally when we're talking about AI. Can this all be done in a half an hour?
It can all be done in half an hour, Adam.
I think the team will laugh a little bit about that, and we might have to explain when we get to our special guest where the inside joke is. But yes, utilizing AI and trying to get macros and efficiency, and I remember you mentioning several times, so this should be done in a half an hour, and sure enough, it's 90% of it can get done in about a half an hour, but there's that last 10% usually that goes a bit further. So glad to have some fun with that.
Right. And that's always it. And the paper does highlight the importance of validation here. And as you say, that last 10% can sometimes take up, 80 to 90% of the time as well. In fact, in terms of software coding, they frequently talk about the fact that although AI can make things a lot faster in terms of generating code, sometimes it can take even longer for someone to go through and debug it because they have to get more familiarized with the software than they otherwise would if they wrote it themselves. All right. And then just continuing the extremely sort of meaty trend here in terms of very dense reads and very high impact for the industry. The first bit of investment news doesn't have any dollar signs attached to it, but is related to how the FDA evaluates biosimilar testing.
So you say it doesn't have dollar signs attached to it. However, I would say that it is very impactful financially, bringing down the aim, in part at least, is to bring down the cost of biosimilars. I certainly am very keenly interested in this part because I had worked for Centocor previously, so you may know for Remicade, and then was became part of J&J. And so, that then, how do you generate a biosimilar? And then what are the requirements here for the FDA to be able to approve such a therapy? And so clinical efficacy studies have been the rate limiting step here for biosimilar generation, and essentially the cost of it. You were setting up clinical studies that would require demonstration of key markers that were very similar to what was seen for the original therapeutic. And here, a biosimilar for Stelara does not need that per the FDA. And some of the background on it is they need to demonstrate that the molecule is essentially like for like. And so with that, if they're able to go this pathway, the article was talking about reducing biosimilar development costs by over 90% and then accelerate approval timelines by 70%. So at IP expiration, essentially then these biosimilars are able to enter the game and offer something that theoretically should perform at the same level, but at a much lower cost. And these types of therapies can tend to be very expensive. Now, what I was talking about as far as my interest here was how do you generate a biosimilar? Because having my experience in Centocor and going through construct generation and then going through cell line generation, there's manufacturing that needs to go into it, a lot of information to be able to create one of these. And essentially what you have to do is reverse engineer, right? There's IP out there, so there is some information. But again, these are biologics. And so, for example, the construct is going to be different or potentially different. The cell line is potentially different. Post-translational modification would then be different based on the difference of the cell line. And so all of this has to be accounted for and controlled for to get a biosimilar approved and out on the market.
That's it, right? So there's still a lot of controls in place and a lot of evaluation. They mentioned several times that the importance of the analytical characterization and the importance of the immunogenicity assessment. However, waiving this efficacy is, the biggest hurdle towards getting the drug approved. Right now, there are very, very few biosimilars that get produced for biologics, right? It is just a massively expensive undertaking, and that stopped a lot of people from going forward. And, as you say, the first step is also extremely important, right? Like, how would you even plan to do it? But with this change in rules, it does open the playing field. So now people can, instead of needing to go after markets that are greater than basically 200 million, because that's the sort of table stakes to get into biosimilars previously, now they can go into much smaller markets, in the 30 to $50 million range, and still potentially make a profit.
Right. Open up the doors for smaller companies that could potentially do it instead of the much larger companies that historically had.
That's it. So it'll be interesting to see how many companies take the FDA up on this and what the reception of the industry is. But I think, when you're faced with a disease that needs a biologic therapy in the first place, then you're talking about a pretty expensive disease. And if you have an option to spend, a third of the price or a quarter of the price, then likely I'm imagining patients would be interested.
Yeah, from a patient perspective, I would imagine that the reception would be very warm. I was interested to see that the US in this regard was actually coming towards the EU, UK on this sort of approval process. And so there was a lot of push to make this the path from some key players within the US to align there. And I guess my perspective was just knowing how much goes into the cell line generation, the manufacturing, and all of that, that there would be possibilities for differences there. But evidently, with the data that they're seeing here, the need for the clinical studies to demonstrate that efficacy and safety are not quite as high, because you've already demonstrated then that the molecule itself is similar. Now there are two versions, right? Talk about a biosimilar or an interchangeable biologic, which you would imagine one would be much more towards the exactness of the original molecule. But really interesting stuff there.
Right. Absolutely. But the FDA invested $0 in that new company. So, $0 invested there. Now, in our next story, of course, Genmab invested significantly more in Mirus Therapeutics. So here they paid $8 billion with a B for Mirus, primarily because of a new bispecific antibody that they have produced.
I noticed you didn't say the name, and I was practicing it beforehand. I always want to try to say these mAbs correctly. The petosemtamab, so demonstrates 79% 12-month survival rate. This is for PD-L1 positive recurrent metastatic head and neck squamous cell carcinoma. And expected to the market in 2027, right? And some of the discussion around here was Genmab looking to build their own pipeline. They've had partnerships in the past with J&J and BioNTech. And with this therapy alone, they're looking at yearly revenue of $1 billion plus. And so very large therapeutic as far as revenue. What is the interest here? It targets, it's a bispecific, so targeting EGFR and LGR5. So EGFR is well vetted in this area, in this space. It's a tumor-associated antigen. LGR5 is highly expressed in cancer cells and serves as a cancer stem cell marker. Interestingly, when you look up LGR5, it's got the quite a few names associated with it, and leucine-rich repeat containing G protein coupled receptor 5. There's several other names to it, but some of the important aspects behind this. There have been 14 EGFR targeted agents that have been approved for cancer therapy as of 2023. So this is coming from a paper that I found in a publication called Expert Opinion on Therapeutic Targets. It's the paper is called Targeting the EGFR Signaling Pathway in Cancer Therapy, What's New in 2023. So as that indicates, this is information as of 2023. Interestingly, EGFR therapy, blockade therapy alone, generally leads to resistance. And so here, the combination of the EGFR plus the LGR5, LGR5 should lead to degradation of the EGFR and should enhance the effect and then mitigate resistance. And so, this combination then is demonstrating great efficacy, which obviously led to the purchase.
That's it. And sorry. That is always the promise of bispecifics or even trispecifics, right? That with the multiple functional aspects of the molecules, they can carry out just a wide range of functions, some of which could maintain the efficacy of the cells that might be carrying them, or some of them might be basically bringing two targets closer together or triggering some other function related to the cells. So sort of a programmable piece there. And with the number of functional monoclonal antibodies that are out and available, it gives you a wide range of things that you sort of already know a little bit about the safety profile. So it's a little bit similar to ADCs, where each ADC really stands as almost a platform. You can think about a monoclonal standing as a platform for different bispecifics now as well.
This was fun because I remember back in the day when I was at J&J, one of the VPs there had a whole slide on the multi-specifics, right? Not just bi-specific, tri-specific, quad-specific. It seemed like you were slapping modalities all over the place on antibodies to be able to bring things closer together. And it's not just antibodies. You had the SCFVs, whatnot, that were also in use, various sizes, various half-lives, so on and so forth. And so now all of these really coming out, very interesting to your point, that how you can attack various diseases.
That's it. Yeah. So exciting news, exciting for Genmab to expand its oncology pipeline, and exciting for Mirus to get a partner to ideally supercharge some of its efforts on development in the near future here. Of course, I was going to move on directly to KCAS News, but then some, little prize in Europe mentioned that, basically selected their winners. And this was actually the Nobel Prize, of course, and they did select the discovery of Treg cells as this year's winner for the Nobel Prize in medicine, physiology or medicine.
Wow, so you're really going hot off the press news, because that was was just today. I know I was looking at speculation on who was going to win which prizes there. So yeah, you said T cell exhaustion. So tell me a little bit more about the release there.
Yeah, so in this case, Mary Brunkow, Fred Ramsdell, and Shimon Sakaguchi won the Nobel Prize for discovering basically peripheral immune tolerance. And that's basically how the immune system knows not to attack your own body. This was huge in terms of understanding mechanisms that we could tune either way for cancer and autoimmune disease. And for cancer, the attempt is really to tune up the response. That way, the body attacks more cells that look sort of similar to the body, because, of course, cancer looks pretty similar to the body itself. And in autoimmune diseases, the thought is, of course, to tune down that response, to basically train the immune system that that's not a robber, that's just your brother, and they're going to get a drink from the refrigerator, so don't attack them in the middle of the night. And right, got to anthropomorphize these T cells. That is the key. And yeah, so this is really exciting. Of course, we know and love Treg cells, and we've grown up with them. I can't imagine working in flow cytometry without thinking about Treg cells and how to find them and understand what they're doing. But this is the group that identified them. And they thought basically that something had to be controlling this T cell response, and they came up with an elegant set of experiments to isolate and identify them.
I love that you're anthropomorphizing there, and specifically the T cells, because I remember one of my immunology courses, my professor, she had put on the slide for cytotoxic T cells, the Incredible Hulk smashing things. Yeah, very, very good. It helps keep it in your mind, right? You learn that way, and I obviously haven't forgotten about that. So very obviously mnemonic devices as well work the same, and I'm totally going off script there, but not that we are on a script anyways. With tuning, I think that's very important, and we've talked about in flow cytometry looking at cells with, for example, high, medium, and low when we're sorting, and an aim for use in tuning responses. And again, I think going back to your point about the Nobel Prize, certainly that's very warranted prize there, and certainly impactful within immunotherapy, cancer therapy.
Absolutely. Also, hopefully impactful within the flow cytometry community for this year, for this month's KCAS News. I've got that we are, and by we, the AAPS flow cytometry group is slowly starting to target the AAPS NBC conference.
Hmm.
And
Now, I think you forgot something though. Like you're traveling too, and you were just at a conference. And you didn't even mention it
that's true. I mean, that's all
in the
old news now,
agenda.
right? Yeah, that's true, right? So we did go to the Festival of Biologics last week. We did go to ICCS last week. We'll be at Nordic Life Science Days next week. So it is conference season. Conference season is in full swing and is causing a lot of travel. But this is fall conference season. So I'm talking now about spring conference season and trying to drum up excitement and support for a new conference. A lot of flow cytometrists will go to AAPS WRIB and they'll go to Cyto and sometimes ICCS as well. Here, we're trying to have another AAPS conference with a lot of flow cytometry talks at it. So the NBC conference, the National Biotechnology Conference, should be another good opportunity for us all to meet up, talk about flow and best practices as it relates to ongoing clinical trials here.
Uh, Going back to the Festival of Biologics, you and I haven't even had a chance to discuss. I'm curious what some of your key takeaways were there.
Yeah, the Festival of Biologics is always very interested in antibodies, in bispecifics and trispecifics and things like that. And they're actually that's the meeting where I got the hot tip about biosimilars and the importance of it. And definitely, as that article said, the EMA has been pushing this pathway for a while, and now the FDA has sort of agreed that that's the right path forward. So there was a lot of excitement and happiness at the conference because now the international regulations are more aligned. So it should allow sort of more entry into that space.
And were there any specific flow cytometry takeaways from that?
Um, in terms of flow cytometry takeaways, there is less focus on the exact analytical techniques and more focus on the construction of the molecules themselves. So a lot of
Mm.
really in-depth discussion on um, the types of bispecifics, the types of antibody modifications that can be made to tune immunogenicity, to tune functionality, to talk about the best types of targets to go after, and where fields are crowded or not crowded, and what can really set different therapies apart there.
So
Um,
very much tangential to what we do. We support the things that are going on there.
That's exactly it, right? Like we go there to talk to our sponsors who are interested in understanding where their next assets are or where their drug that they're developing may fit into the landscape. And at the same time, they can talk to some CROs about how they can get to the market faster as part of that.
Excellent. Sounds like definitely well worth the time.
Yeah, no, it's a great conference always. Always good to be over in Basel and gives me a good excuse to go visit our lab in Lyon, of course. Yeah, and then maybe we can move on into the paper of the month and our main topic for this month as well. And to do
Sounds
that,
good.
also we'd like to introduce uh, David a little bit in a little bit more detail than he did on the intro. So, David, do you want to give a brief introduction to our listeners?
Yeah, absolutely. So I am director of the CAT facility at the University of Chicago. CAT is the Cytometry and Antibody Technology. We essentially serve our research community in all things flow cytometry. I've been here for 20 years this year. I'm one of these people who find a job and decide never to leave it. And it's just a fantastic position here. Great research community, and the administration provides tremendous support. So I can only wish that kind of position to any one of my colleagues. Um, did my master's in Montreal, Didn't like it much. It went terribly wrong, and so basically ended up in a core facility in Ottawa, and from there in Chicago. And you just stuck around long enough, and you become leader of everything here. Um, and yeah, so that's about it. I'm also in my spare time. I've been leading the effort to bring GLIFCA in Chicago. GLIFCA is the Great Lake International Imaging and Flow Cytometry Association. We do a yearly meeting. Typically, we end up in places like Troy, Michigan, and Oconomowoc. And this year, we decided for the very first year to be in Chicago. That turned out to be a great meeting. Um, so yeah.
So, David, I'm really curious because listening to one of your podcasts recently, this was the one from August, I think you had mentioned about your master's experience and it basically characterized it same as you just did there. Curious if you had any stories that you wanted to share, something, even obviously a lot of it was flow cytometry, from my understanding. And then what has kept you at the job? What is the primary interest there at your job?
Well, yeah, so it was a difficult project. I'm not going to blame anybody on why it went so poorly. I think the main reason was I did not know what I was doing. But the big part of the issue was the we were working on the impact of hexachlorobenzene on the basically cancer development down the line, and how the exposure to hexachlorobenzene would impact that process. And the issue was that it would take about 3 months to set up our rats so that they develop the characteristics that we wanted to study. And so you would basically prepare your rat for 3 months and then get to the experiment where you need to harvest the tissues and something would go wrong. And then you lose the tissue and then you wait again for another 3 months. And so it was just tedious, boring work. And I didn't like it much. Um, I did awful flow data over there. I have nothing good to say about my master's. It was not a good, happy place in my life. I learned how bad my flow data was when I actually got a job later on in a core facility, and I went, "Oh, that's what I should have done." So, yeah, um, no, but the flow part was the most fun because I got to move like the instrument available to us were basically in a different campus. So if you know Montreal, it's an island. Actually, there's an island called Montreal. There's a smaller island called Laval just on north of it, and I needed to move from one campus where the most of my activities were to a different campus in on that second island in Laval, and that's where the machine I was using was located. And you would go there, and there was a core facility director over there, Marcel Desrosiers is his name, and he would basically every time I showed up, he would just pop in and start talking about the flow stometry world. And at that time, he would just basically have stories about the different companies. At that time, he was talking about how cytomation was just coming up on the market with the MoFlow, just like taking all over the world with their new instruments and leaving behind the people on both of the coasts. And it would just talk for hours on end. And so that just gave me a big just wanted to stay in that field.
Mhm. Yeah, certainly appreciate that. And then moving on to your role now, mention about technology. I know that ties in very well. Obviously, Brian had planned this. He, plans everything. That's what he does. But specifically, the paper is Flow Cytometry: The Next Revolution, and so it's published in Cells. Impact factor of 5.2. This is by Robinson and team there. Curious as to your thoughts when you hear flow cytometry in the next revolution.
Yeah, I was very surprised about this paper. I deeply appreciate the perspective of J. Paul Robinson. I think I can recognize the parts in the paper where that he wrote personally. Whenever he talks about the history of flow stometry, it's always quite entertaining and insightful. I I was kind of surprised to see that the microfluidics is where they're going with as the next wave of flow cytometers.
Hmm.
I kind of we can discuss about being a purist myself. I don't know if I'd consider that flow cytometry per se, um, but
Now tell me why.
I well, it's basically in this case we're just encapsulating the cells. It's just a different ways of manipulating the cells and doing something with them.
Yeah.
But we don't have lasers. We don't have the general tools that we don't have compensation. If you don't have compensation, you're not doing well, or in mixing, you're not really doing flow cytometry, are you?
Now, what about if you're doing compensation and unmixing?
Oh yeah, no, that's a fun question. But I don't know. So I kind of get the point though. I think I if I would reframe, and I don't want to put words in their mouths, I would say one of the nice, very 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 nice advancement in flow cytometry has been our ability to manipulate the cells in a different way than this droplet based cell sorting
Hmm.
technology that just basically took over the second it was developed because it's so good, it's so fast, it's so incredible. But nowadays, there's a bunch of different ways that allows us to move the cells around in ways that are just so much nicer. Granted, the microfluidics encapsulation is great. They mentioned the Berker. What's the name of the platform? Um,
Is that the Molotov?
sorry, it's the Berkshire. Basically, they have this platform that allows you to move the cells around and move them to a specific little hub where they're going to get treated, and then you can measure the expression of the cells, different analytes, and then figure out which of the cells respond the way you want, and basically take these cells, move them to the exit port, and then keep these cells and continue your technology. You're not doing that through encapsulation. You're doing that through electric field and light signals. And so that's another way we're dealing with the cells. If you know anything about me, you know I can't shut up about the Max Quant Tido from Miltony. That's another way where cell sorting has now kind of shifted gears, and we can now uh, basically sort just as fast using the side speed valve that you find in the cartridge of this particular instruments this particular instrument uses, and basically sort in a way that you don't worry about contaminating your sample. You don't worry about any kind of biosafety issues. You're just basically uh, collecting cells at high speed uh, and basically keeping them as happy as possible. All of these different things are now available to us, and just like fantastic uh, improvement on the quality of the work that's going to get done going forward.
I was interested to hear you mention about the microfluidics. The droplet microfluidics interested me because, as mentioned about being back at J&J and doing stable cell line generation, one of the things that we were trying to do was look for an alternative. A lot of the stable cell lines were picked using a robot called the clone picks, and you
Mm.
basically seed the cells in a semi-solid matrix, and then you're looking for fluorescing colonies, and then you're picking them, and then you're expanding. And what we were trying to do was shorten that timeline and try to sort on cells. And so we were staining the CHO cells, but it didn't necessarily correlate the intensity of the fluorescence on the surface of the cell to which were then downstream the highest expressors. And so that's what the clone picks would typically do really well, was each one of those colonies, the ones that were higher expressing, they would express higher amounts of antibody within that semi-solid matrix, and that would be contained. And so here, utilizing droplet microfluidics, I would imagine that that would certainly be an application. Then they do mention stable cell lines for high protein producers, and so certainly see the value there.
Oh yeah, yeah. I'm not familiar with that platform. Is it image based or is it just again more flow cytometry based? platform?
So the clone pix actually isn't flow cytometry based. It's more just camera
Yeah.
taking a picture of a plate and looking at colonies. I was using a BD Fax Jazz at the time to do that. So if you're familiar, it sounds like you are familiar with that instrument. Very non-automated, and so that's how I learned about aligning lasers and droplet delay and all of that kind of stuff. Really was fun to cut my teeth there.
I wanted to make a point. I'm laughing because I recognize the instrument, not because I'm laughing at the instrument.
No, understood. I certainly
We did
appreciate
good.
it. I think it was a
Yeah.
fantastic tool. I know what you mean.
Yeah.
It was the engineer's cell sorter, definitely.
Mhm.
All things adjustable manually. So definitely good there. Yeah, and one of the things that I wanted to bring out about this paper was just around, as you said, David, how cells get from one place to the other. Certainly, with cell sorting, it becomes extremely important, as Adam says, being able to maximize both the yield, so the speed at which you can sort cells and how happy they are at the end of that process, while not destroying the cells by going too fast, is a difficult balance to hit. And as you say, we've made excellent strides there. But even in analytical flow cytometry, where we're not actually sorting cells, but we're just running cells through the flow cytometer and looking at them, being able to go faster, definitely from a CRO perspective, is a huge benefit, but also has some drawbacks to it. I think what this paper
Oh yeah,
really
totally.
mentioned was, of course, it
Yeah.
shortens the amount of time that the cell's in the interrogation chamber, and can lead to some reduced sensitivity in some cases there.
No, yeah, so it's a question that is very often uh, glossed through in an introduction to flow cytometry course. Mine has one slide on it, and then we kind of forget about it for the rest of the presentation. And people are very often deciding on the pressure they're going to apply to their sample based on how bored they are in front of the instrument, where really they should be caring a little bit more about the quality of the data.
Yeah, it's
Exactly.
not only how bored, it may be how late they are in the lab while they're working on their PhD and it's 1 o'clock in the morning and they're just really trying to get out of. So I may be talking from personal experience, but yes, you've got that high, medium, and low setting, and there's always that temptation to hit high. I typically was slapped on the wrist by biology, though, in doing that because my PhD work had to deal with macrophages. And so going fast invariably clumped up the cells and clogged up the lines, and so then I had to back it off. I had to clean it up, and yeah, so that was my experience there.
Oh yeah, it strikes me that you guys are more in industrial context, and there's definitely a question about the throughput that you need to reach to run through all your samples. You might have to look into if you're screening something, for example. You might not have all the time in the world to run your samples. It might actually
Mm-hmm.
crash before you run through your plate or something. So
Yeah, it's good. You've got to be efficient too, right?
right,
Because clients aren't going to pay you to sit on your machine for hours running samples, and so the faster you could get that done, the cheaper it's going to be for the client. And you don't, but nobody wants to sacrifice quality, right?
right,
And if you go too fast, then all of a sudden there are questions about quality. Uh, Obviously, when we're talking about sorting, you're going to have a lot more aborts as you increase the flow rate there, and it's certainly it's something to keep in mind.
yeah. So
That's
how
it.
did you want to do this? Do you want to go into how what's the impact of the pressure on the sample? Because in a few words, like I feel like it might be a bit obvious to everybody, but as you increase the pressure, you're essentially increasing the diameter of your sample core within the stream of sheet. So whenever you include you run your sample in the instrument, the sample gets focused by a process called hydrodynamic focusing, which essentially you squeeze out the space the cells have to navigate in front of the laser beam with a bunch of sheet to to a simple, very narrow uh, core where the cells have so little space to run one to navigate that they're going to be essentially forced to run one after the other. And since that simple core is so fixed and so stable, you can now align the lasers to get the best possible illumination that you're going to get as the cell crosses the laser beam. Now, as you increase the pressure, you increase the diameter of that simple core. Now cells have more space to move around, and they're not going to go necessarily in that focus point. And now you increase the variance of illumination of each cells. And on a graph, when you plot the data, you basically realize that the cell starts taking more and more space on your axis, and basically real estate that in some particular case might be highly necessary to separate different subset of cells that have really similar levels of intensity, right? And so these assay would be something like anything if you're doing phenotyping and you're trying to look at really dim markers, low expression. You expect your signal to be very, very low, very similar to your autofluorescence. That's going to be a bit of an issue. If you're doing complex spectral flow cytometry, really high number of markers, the lower the pressure, the more on target your signals are going to be. The better you're going to be off resolving the different signals. If you're doing cell cycles, same thing happens. So yeah, there's a bunch of assays like that where running at low pressure becomes a critical thing to do.
That's it. And for high pressure, you need to make sure that your populations are bivariate and with a pretty significant shift in between the positives and negatives. And then as long as that shift is significant enough that if you all of a sudden double your CVs of your assay by running it too fast, then it's potentially justifiable, but definitely with great caution and with great validation beforehand.
Absolutely, yeah.
And I guess one last sort of technical piece that David, you had actually mentioned in terms of the run up to this podcast is around acoustic focusing.
Right. I mean, so this is it's actually I saw this technology the first time when I went to the annual flow course. It was given in Los Alamos back in the days. That was 2006 or so. But essentially, the general idea is you can use sound waves to align the particles in a straight line before you introduce your sample in the stream of sheet. And so even if you increase your pressure on your sample, the cells are already aligned. And so even if you increase the pressure, you increase the diameter of the sample core, the cells do not navigate anywhere else than where they're already set up by that acoustic focusing. And so that allows you so Thermo Fisher offers the Attune NXT, the Cytopix, and now their spectral platform is the Zenith, and they all have this acoustic focusing. So essentially, you can run 10 times faster, give or take, than you would on a traditional instrument, and still get somewhat the same quality of data that you would otherwise get if you were running at lower pressure on other platforms.
Yes, and so we in the Philadelphia area have a new group called MAFCA. So it's the Mid-Atlantic Flow Cytometry Association. We just did a demo with the Cytopix, and definitely a lot of people were very impressed with how quickly the instrument could run. Certainly, then the limitation shifts from being fluidics and fluidic stability over to data acquisition speeds, right? And specifically on the Cytopix, they've got one speed, I guess, for lasers and standard detector arrays, and then another speed for image. Because, of course, when you're processing that many images, then you can only do that so fast, even with our modern processors and things like that. But very cool technology overall, and anything that can help us go much faster and screen cells and much better than we're all for, as Adam says. Like the client only wants to have us run the minimum amount possible for acceptable quality. That is always the goal.
Yeah.
And maybe that's a good transition into the sort of second part of our main discussion here in terms of the differences, some of the differences between academic flow and flow cytometry as practiced in industry and specifically in CRO settings here.
Yeah, so David, really curious from your perspective. I know you've been at the University of Chicago now. You said 20 years, right? And so you've, I'm sure, collaborated with CROs, big pharma. What are some of your thoughts there?
It's actually it's actually the opposite. I don't work much. I don't feel like there's a whole lot of communication between academic world and the in terms of core facility, at least between the academic and industry.
Hmm.
And it kind of, I noticed something this year when we're organized GLIFCA in Chicago, where we made a point of trying to incorporate the point of views of there's a lot of biotech developing around town, and so we try to invite them and present at our in our facility in our meeting, and there's a bunch of things that are done in industry that I think if we knew more about it, the core in academic settings would definitely benefit from them. One of them is definitely automation and sample preparation. We had one talk by Ryan Duggan at AbbVie that was pretty phenomenal, just like how he prepares the samples in a way that makes the quality of the data so more reliable, more like reproducible. All these different things that are really cool. And on the other hand, I'm not sure where what I was kind of curious is, since I don't talk to anybody in industry, I kind of wanted to know, where do people working in industry keep their, or how do they keep fresh with the new technologies, and how do they keep aware of like what kind of instruments come in, and how does it work, and how can it benefit their work, and so on.
I'd definitely say that, going to your last question there, we stay on top of that, similar to the way that academia would, is going to conferences,
Right.
connecting with colleagues there and looking at what's being offered. There's always a lot of excitement. We send team members out. Brian had mentioned ICCS, Cyto, WRIB, really getting out there to look at what's available. The team wants to see the new technology. Now, I would say from a CRO space, we're not going to be at the very cutting edge of adopting a technology. We wanted to adopt spectral flow since 2017, but there's a lot of time the industry needs to cut its teeth on such things. And so we certainly maintained awareness of it. And then as it became more widely adopted, we brought it in and, essentially adopted it ourselves. So I would say it's interesting there. I guess having in my past, I was always thinking about the bigger industry adopting technology really fast, but I think big pharma tends to do that more rapidly than do CROs. And I think where do you say, as far as adoption, academia and getting the new toys there?
I think it's just generally the same idea. Like if you're a smaller core with like funds that comes every other year for new equipment, you're going to be much more conservative in what you're going to be buying.
Hmm.
But if whenever the money is widely available, then you might be um, more eager to try the newest platform and the newest tools that will provide the data to write the papers in Nature and whatnot. So yeah, there's a range there too, that at the speed where people accept the technology.
It's interesting too. Your podcast spurred some discussion among my team because I had shared it with some members of my team this morning, and to talking about the discussion on reference controls and on mixing, and how I couldn't imagine having adopted spectral back in 2017 and running clinical samples at a
Right.
time when really there's still so much to learn. There's still so much to learn now.
Yes.
Obviously, there's some disagreement on the what was the compensation on top of unmixing and what is appropriate, what's not. No, Florian had mentioned about not having negative values for MFIs and that indicating improper unmixing. But then we were talking about plus or minus really at the edges, these very small changes. And so frankly, I'm kind of thankful that we weren't at the very cutting edge because we would have had to try to answer questions that at that time we really would not have been prepared for.
Right, and it's not exactly benefiting your bottom line, right?
Right.
You just
And then go ahead.
No, I was about to say, like you mentioned the podcasting, and then this is based on the idea that if you're gonna if you're gonna attend meetings in order to keep track of what's going on, I think the idea of podcasting and the idea of meeting online is also something that's going to probably go develop over the years. That general ideas, I feel like meetings are getting expensive, and we live in strange days where actually attending a meeting might be very difficult. I think these online discussions are going to just become more and more interesting and more useful for a wider group of people that might otherwise not be able to attend those meetings.
And in that podcast, there was a mention about where do we centralize information. There are some key antibody interactions. For example, I forget what the fluorochrome was, that when it's actually binding on monocytes, for example, you start seeing degradation, whereas you're not seeing it if you use it on other cells. Apologies, I'm just blanking on. Do you remember off the top of your head?
No, not really, but I know what you're talking about. I know the time in the discussion. Yeah,
right. And so specifically there, how do we collect that information? Because a lot of people will bump their head on information like that, and that's how they learn. But there's no reason why each person has to do that.
Right. I think that's why we need, like, I'm not sure if you're aware of like groups like the Flowstometry Discord group or Reddit have developed quite intensely over the last couple of years. And this is basically places where you see all these experts in flowstometry kind of asking these kinds of questions and very quickly getting answers. I don't know that there's a fun way to keep all these weird facts about what antibodies work and what order with it. But if you can at least get access to the people who know, and then you get the information, and then it basically lives around this flowstometry community brain that we can develop on these platforms, then that at least is one place to start, I think.
Yeah, I know Brian's been participating on some of that, the similar, efforts to standardize. Talked about SoulCap on the podcast, right. And so, Ryan Brinkman, just really good effort there. Brian, anything that you wanted to add there?
No, I agree that it is a common question, right? And the theoretical best place to centralize all that is some type of database that would be curated and maintained by ISAC or some other type of body that has sort of widely accepted by the field, right? Otherwise, you get the feedback of definitely experts, but potentially without data backing it up. And if it was centralized in some sort of database, then it might be more reliable or more searchable or easier to find. So I think it has to develop as like a groundswell sort of organic effort. And then once it gets to a certain level of engagement, should then migrate from more of a grassroots type effort into a sort of systemic tool or resource for the wider community. But agree that there's just so much that you have to explore with Spectral that there's no other way to do it aside from asking questions and getting responses, much like what the old coding databases and things like that. So,
yeah.
All right. I know we are moving on in time, so I will maybe transition over to the feedback request, which is also sort of similar to one of our main topics, and that feedback request, what we'd like all of our listeners to respond to is, what surprised you the most when you got your first position in industry? And Adam, when you joined Centicor, I suppose, what was surprising to you when you joined there?
I will say my first industry position was actually with Purdue Pharma. I had gone from academia. I was in Albert Einstein College of Medicine, the Marion Besson Liver Research Center up in Bronx, and then went to Purdue Pharma. And I would say the biggest thing that surprised me about industry, and looking back on it, probably shouldn't have been a surprise, is just the fact that you aren't bringing projects to completion more often than not, especially as a scientist on the bench. You're probably going to be moved from one project to another. Project's probably going to get canned or, move on to a different stage. And so I had one of my boss at the time actually said, don't get attached to the project. You got to be flexible. You're probably going to be working on something else in, couple weeks. So just to give you a heads up. So that was definitely a surprise for me. How about you?
Yeah, I went straight from academia into a CLIA certified laboratory, and just the amount of documentation and the tracking of that documentation, and the intense focus on documentation as actually being as important, if not more important, than the science itself, was very surprising to me to see coming from an academic background.
I totally get that. I would say most of my experience prior to coming here was with discovery, and so here, I mean, the impact, the driving for documentation, obviously, is very high. In discovery, less so, obviously, for specific reasons. But yeah, I understand what you're talking about.
Sure. And David, what about the switch from masters to core lab work? What was most surprising there?
Oh, it was. I don't remember my days in Ottawa in Canada. That was far away. But like I remember when I got in Chicago, I was very surprised at the level at which we'd be able to play with the instrument, try to fix stuff on our own. I always working in institutions where they paid big bucks for these instruments, and nobody but the service engineer would be able to touch them. And then you get in Chicago, and then suddenly we're like opening computers and instruments and realigning lasers, and nothing seems to be too far fetched for our reach. So that was a lot of fun. I think going in industry, I think the HR, the quality, how people judge the quality of the work is what would be the probably the thing that would get to me the most. Like here in Chicago at the university, uh, we do self-evaluation, and everybody is pretty happy all the time with the work I do. I think once you're in industry and you have actual goals to reach, that probably will change things a bit.
Sure. Yeah. No. Definitely. The annual goals process is always intimidating there. All right, and then sort of moving into, I think, the last section of our talk here today, just in terms of general banter. What undergraduate institution did you attend, and how did you pick that specific undergraduate institution?
I think it's great that that was your question. I had totally spaced on the fact that that was going to be the general banter, and I led off with that with your first article. So I'll just take this at the outset. As I mentioned, I went to Penn State. Absolutely loved my time there. It's such a beautiful university. Now, I grew up in New York City, in Queens, and so I had the opportunity to either go there or stay home and go to St. John's. I had, some scholarship or something. Parents were trying to convince me to stay home. My brother-in-law goes, I've got friends that they went away and they said it was the experience of their life and they wouldn't change it for the world, and kind of sold me on that. And also knowing the kind of institution that Penn State was, is like, yeah, I'm going away. And I would say for myself, that definitely helped me mature. I remember a friend teaching me how to do laundry within the first month, and getting lost on campus and whatnot. So certainly changed who I am significantly. How about you, David?
I did my bachelor at Laval University in Quebec City, and that was a lot of fun. It was basically, I come from Quebec City, so I stayed with my parents during that time. Yeah, I just love biology. It was fun thing to learn.
Brian?
Fair. Yeah, I went to Vanderbilt University in Nashville, and chose that after a long screening process where I insisted that I try the turkey sandwich from their cafeteria at all the potential academic institutions I would go to, thinking that I would probably eat turkey sandwiches relatively frequently during my time there, and I chose the one with the best turkey sandwich. So it was definitely a good experience, and found some good mentors aside from the turkey sandwich piece as well.
I would say I never would have expected that story. Now, do you still like turkey sandwiches, or did you burn out on it?
I burned out hard. No, I will not eat them anymore. Like there were too many at the
That
time.
was just
So,
your favorite then, or what?
yeah, that was it. Yeah, at that time. So this is why you shouldn't allow high school students to choose their futures. Like this is too much for their tiny developing brains. So I don't know. For me at least, definitely.
No, mine. My story kind of mirrors. I could have put a lot more thought into programs and all of that kind of stuff, but at the end of the day, it was my brother-in-law convincing me that going away was the best interest for me. So at least it turned out it worked out well. So yeah.
Nice. All right. Well, again, thank you for all of our listeners. Thank you, David, for joining us. This has been a great discussion. Appreciate the discourse here. And looking forward to all of our listeners joining us next time for our 26th episode.
Thank you, everyone. And thank you, David. Really appreciated having you on.
Thank you.